All right, we're starting a section on DNA biotechnology, and I'm going to introduce things by going over some basic tools that we use and talk about some of the processes that we do. Particularly, I'll focus on recombinant DNA and talk about how genes are copied, sequenced, cut, pasted, things like that. And so the first picture here I had was one of the early uh, biotechnology successes. It was cloning, where we could take cells from an adult animal and transfer the nucleus into an egg cell, and do that by causing those cells, removing the old nucleus of the egg cell and putting in the new nucleus and fusing the cells together. And cloning of animals was one of the first big steps that we saw. Uh, not hard to do, very easy to do. So let's clear up some things. There's a lot of terminology and jargon here, but it's the ideas are kind of simple. Biotechnology is just using living organisms to do something. Um, it's doing something, making a product. Genetic engineering, genetic things like that. It's using DNA to do something. When they say transgenic or genic anything that's making it with DNA, um, all of this stuff was originally done in test tubes. Much of it now is automated, very easy to do. Um, Even when it was done with test tubes, it was mostly done pipetting, different types of solutions. And this came about from our understanding of cell biology and the chemistry of cells. Particularly, people started recognizing that enzymes were very specific, that they controlled specific chemical reactions, taking products and making them into substrates and being reusable. And they did this reliably, whether they were in cells or outside of cells. And as we got to know how DNA was handled in cells, we recognized that DNA could be cut, pasted together, or healed, copied by enzymes that cells used, or that were purified from cells and viruses. And these could be get to work in a test tubes. Uh, and we gradually began to understand that bacteria and viruses didn't use meiosis or mitosis, but they did have mechanisms for copying and inserting genes into cells. And so this here, I have a basic picture of an enzyme where an enzyme has a shape that matches a substrate, and then it converts that substrate into a product. That's true when that's DNA, and those enzymes work as long as they have substrates available and the right pH and the right temperature. And so a lot of this stuff comes from understanding bacteria and viruses and that how they handle DNA, how they can transfer genes. And we recognize that DNA can be picked up by a bacteria from its environment and inserted into its chromosome. And that there are some types of DNA, like molecules called plasmids, that are specially designed for this. And so we gradually learned that ways that DNA and viruses transfer, and we learned to stimulate those things or get these things to happen when we wanted to. And gradually, it, people realized that DNA was found in all cells. It was the inherited material for all cells. All cells use the same processes to convert DNA into RNA into proteins. They use the same genetic code. They use the same amino acids. And so they use the same enzymes to copy and handle DNA and RNA and things like that. And so once they knew those enzymes and they could purify them, they found that they worked in test tubes. And this was the basis for DNA technology. In addition, uh, we also had enzymes that we got from bacteria that could cut DNA at very specific sequences. Um, these and or recognize specific sequences and bind to them. These are DNA probes and enzymes that recognize particular base sequences, so they'll match that sequence. Some of these, as I mentioned, cut DNA. These are restriction enzymes, And so they find a certain segment on the DNA molecule that has the right sequence and they cut it there. Other enzymes can reheal those cuts. And so that gave us the tools we needed to identify specific molecules of DNA, cut them in specific places, and reheal them. And we could put DNA molecules into those plasmids or other molecules that are normally inserted into cells. So once we made these things, we could get cells to take them up. This is the basis of genetic engineering. Cutting a piece of DNA, inserting a gene that we like at a restriction enzyme site, sealing that up, and then transferring it into cells. Okay, those cells will then make those genes into proteins. And so this is genetic engineering. It's really the use of a couple different enzymes to move genes around and put them in different places. Okay. Some of the applications for this have been very, very powerful. We've been able to make medicines that are natural products of the human body, but we can make them in unlimited amounts by moving the genes from human cells into cells we can grow in the lab. So we can make hormone molecules in unlimited amounts. We can make vaccines in unlimited amounts. We can even insert molecules that we want into plants. And so in agriculture, we've made crops that are resistant to particular herbicides that kill all weeds. We've made crops that produce their own insecticide in their own tissues and will kill any insect that feeds on that crop. So this is really, really important, and there's lots of applications. So I thought I'd go over just one today, and that one is the use of insulin, human insulin. In diabetes, a very, very common disease, people either don't produce insulin or they don't produce it in sufficient amounts. Okay, we used to get insulin through purifying it from the pancreas of pigs. This took a lot of work. We would have to get these mixtures of pig parts and go through a chemical separation to remove all contaminants. And there was limited amounts, and so it required a lot handling of lots of things. Instead, what we do now is we've used restriction enzymes to cut human DNA, and we've taken the insulin gene, which we can recognize, and we've pasted into a plasmid. Okay, we've used the enzyme DNA ligase to do that, and we've taken that plasmid and inserted into bacterial cells that we can grow in test tubes or any kind of container in the lab in unlimited amounts as long as we have sugar that we can feed it. What happens then is the bacteria takes that gene for insulin and it makes human insulin. In an aseptic environment where there's only a few other products that we have to separate it from, these bacteria will make this insulin as long as we feed them sugar. So it's a very cheap way to produce very high quality insulin in unlimited amounts. And it's human insulin, not pig insulin. So it's very likely to be accepted by patients and to be very effective in treatment. We do this with lots of proteins that are needed to treat diseases. Blood clotting proteins that are needed to treat hemophilia, growth hormones needed to treat dwarfism or other growth defects. We use an immune protein called interferon to treat cancer, uh, vaccines to alert the immune system to proteins made by infectious diseases, many, many more of these that we can produce. And they're all can be produced inexpensively, very pure human molecules that are easy to separate, very high quality. So we'll talk more about these and more about some applications of these later on. DNA is, um, in summary, DNA is a chemical we find in all cells. We know the enzymes and other cell chemicals that allow us to find and manipulate DNA. These allow us to easily copy DNA samples, identify sequence, and specifically move genes to where we need them. Very, very effective.